This is a production of Cornell University.
So, hi everybody. My name is Siim, and I'm a second-year PhD student at Mark Sorrells lab, working with winter malting barley. And today I'm going to talk about a little bit how to leverage genomic and phenomic data in winter malting barley breeding to predict better grain yield and malting quality. So, as we all do know, that barley has lots of important uses in the world, like feed, food, malting, and distilling. But at the same time, what's most fascinating for me is how it is one of the most widely grown cultivated species itself, ranging with growth areas from southern Finland down to some arid areas in Africa that are pretty deserty. And at the same time, the importance is also here in upstate New York, as the state of New York has the second most craft brewers per state after California. And this has sparked something which is called the Farm Brewer Bill, that is supporting the use of local ingredients in the beers brewed in the state of New York. And at the same time, the New York Department of Ag and Markets leading the initiative with us here at the Cornell Small Grains Lab to breed locally adapted varieties for the New York farmers to have local ingredients for their local beers. The most crucial traits impacting the value and utility of the produced barley for the farmers are malting quality and the grain yield itself. And if we look at those traits through the eyes of the breeder, we kind of see them as there's something that are developed throughout the season, complex and cumulative by the nature. And if I were to sum them up in one kind of phrase or sentence, I would call them a complex production and reallocation of metabolites over the growth season into the grain, which is something determined by the genetic and environmental factors. And I thought that this kind of a visual of the zinc source would be good here to understand how the photosynthesis is kind of producing those things in the plant, and how all of them are finally realized in the grain is also really dependent on the environment itself. So that is really understandable with the yield, but also if we look at some of the malting quality traits we're breeding for, they do sound a little bit complicated, but what they're mostly about is just different kind of proteins at different ratios in the grain that we are aiming to target there. And also some carbohydrates like beta-glucan and some enzymes like alpha amylase. So the breeding for yield has been pretty successful with some of those latest technologies. Like we have genomic prediction, which works with quite a range of different heritabilities and gives us pretty accurate and fast selections, and also more efficient use of resources. But at the same time, when we're breeding traits that are kind of developing throughout the season, we have to keep in mind that the grain yield itself is only something we see in the end of the season. And by putting that into the genomic prediction model, we are not really capturing what's happening throughout the season and this kind of temporal dimension in there, which kind of is missing out on the G by E part there. So one of my ideas is to look at those plants in my breeding program through the lens of a drone with a multispectral camera to kind of be able to capture more what's happening throughout the season, capturing the growth of the plants, the biomass, also the speed of development, and if they're stressed or not. That should hopefully be able to make my predictions better in a breeding program in terms of yield and malting quality. And what I've been thinking about is to try to integrate the genomic data and the phenomic data from the drones to, yes, get even higher prediction accuracies. And the potential benefit of that would be, as an example, helping to cut the cost from harvesting in a breeding program, being able to predict yield already on smaller plants in earlier generations, like in smaller plots, and also potentially improve the prediction accuracy with less related material. That has been a limitation in genomic prediction. But the main economic benefit, or the biggest gain, could be achieved from being able to get good estimates of the breeding values in terms of malting quality. And if we get that out of the model in the same time frame when we harvest the grains, this would enable us to make selections immediately before we sow the seeds again in 2 months, rather than wait for 6 months to get the malting quality data back from the lab. And there has been some success shown using those drone-based prediction models already in maize and wheat. Not so much in barley. Barley has a couple of multivariate genomic prediction models being tested on and being successful, but nothing with the drones specifically yet. So this is kind of the gap I'm trying to aim for. And also to be able to see if this kind of breeding system would be successful with a multispectral drone, which is much more affordable to different kinds of breeding programs compared to the hyperspectral drone imagery. And so this is one of my objectives, to be able to integrate the genomic and phenomic data to get better prediction accuracies in terms of yield and malting quality. And this is the information about the data I'm working with, which is actually drone imagery from 4 years of field experiments, where I have captured the field every 14 days, roughly from April to July. And I'm trying to use the information extracted from those reflectance maps to put it into a model and make a better prediction model. Yeah. So the malting quality has a little asterisk there, because for the malting quality, I have less data, approximately from 2 years, because it's just more expensive to get out. And here are some of the model ideas I'm thinking about testing with my genomic and phenomic data to kind of compare them individually, see how they compare to each other, and also potential approaches to be able to combine them together, and to see different interaction terms like G by E or M by E, where the M is representing the phenomic relationship matrix in a linear mixed model. And another kind of a new approach to look for is a random regression model. That is something that could, as far as I understand, is able to capture the temporal relationship in time better, how the canopy is acting in there. And then after I have all those models worked out, I could be able to test all of these models in different cross-validation systems to kind of help me to point out in which scenarios would be the applications in those models the most optimal. Yeah, here are just the different levels of complexity with some of those models. And yeah, so the cross-validation scenario 1 is what I've been testing right now with some of my preliminary data, and I can show you this to you soon. So this is some of the progress I have been achieving right now. This is a quick overview how the data is being extracted from raw images to the orthomosaic, and being able to extract the mean value of the reflectance from the plot screen displayed on this orthomosaic. And here's a good kind of visual how, like an NDV, something like an NDV value is actually varying throughout the season and also throughout the field. So the point of here is just to show that the sources of variation are coming from different, angles. Like we have the spatial variation, we have the temporal variation, and also we have a variation caused by the genotype, which is something we really want to capture in there. And here's another plot where I just have plotted all those NDVI values on the y-axis, and the x-axis is the time points throughout the season. And it all kind of looks really messy. But on this plot on the right, that's something where I have separated the top 50 lines with the highest yield and bottom 50 lines with the lowest yield. And kind of this gives a kind of a good understanding what's really happening on those canopies of those plants with those NDVI values that kind of already we see that they are determining the yield of their barley plots really relatively early in the season, as the starting point of the plot is in April, which is most likely kind of capturing the winter hardiness of those plants. And so I have put together some prediction models. So this is for the cross-validation scenario 1, where I'm just using the data from the environment to predict the grain yield. And this map is just solely here to be able to compare things like genomic and phenomic prediction. Like, is the phenomic data even worth going after? And these plots all here showed 3 different years I was capturing the data. And so the red model is representing the prediction accuracy of just the spatial correction model. And the beige model is the prediction accuracy of the spatial correction with the genomic prediction model. And the following models are different types of phenomic prediction models, like linear ridge regression, lasso elastic, and one non-parametrical random forest models. And we do see that even though genomic prediction has a success really good over just the spatial correction, almost every year we have the phenomic prediction models doing the same level of prediction accuracy on their own as the genomic prediction, which is something pretty promising, in my opinion, to look into that more. So from those models, I've also extracted something which is called a variable importance scores, that is showing that is aiming to show the time points that are most important in predicting this response. And this has been of my interest just to see that what kind of growth or development of the plant is most important to capture, to see what determines really the yield. So these are also like 3 years here, and the black vertical lines throughout the plots are the mean heading dates of the plots. So we see that the higher the lines, the more important the variable as a time point. So we see that on each of those plots, the highest variable importance score is achieved approximately 10 or 20 days after the heading, which really goes into the flowering and the starting of the grain filling time, indicating that this is one of the most important time points to capture in the crop canopy. And also on those 2 rightmost plots, we do see that the variable importance score is also high in the beginning of the season in April, which is also most likely showing us the winter hardiness and how this is important in predicting the yield. And yes, so here are just couple of bigger questions I'm trying to figure out. Overall, in this data, hopefully I'm also getting the malting quality data in sooner. And yes, so the key questions are like, what time point and vegetation index are the best predictors in any of those models? What kind of growth dynamics do determine the yield? And how does the prediction accuracy vary across different cross-validation scenarios that would be that I could possibly use in a barley breeding program? And here are just a couple of next step I'm working on to finish up the data collection and kind of still work on the development of the methods here. And here I'm just showing a couple of acknowledgements for all those people that have been helping me out working on this project and also with some other projects. And at this point, I do thank you very much. And if you have any questions, I would be happy to hear them. Thank you. Yes, Kelly.
I'm just curious, how are you planning to integrate remote sensing data across years and locations, since they're kind of inevitably captured at different times? Or are you going to treat them as being like nested within each slide, or are you going to try and extrapolate some common time point?
Yeah, so the question was, how am I trying to put together the drone flights that have been conducted at different time points throughout the season in different years? So what my approach is right now is to try to approach that from both ways, kind of use a nested approach. And the other one would be to try to use, I forgot the name of this variable, but it's called the growth degree days that can standardize that, which I haven't shown here. Right here, I just showed the dates of the flights taken. But yes, all of those NDVI values can be standardized to the growth degree days of the specific growth year. Yes. Yes, Ed?
Yeah, you have some very high prediction accuracies there, and I was kind of wondering, you mentioned it related to perhaps survivorship during winter, Was the survivorship during winter relatively Mendelian, or is it very quantitative, or
Yeah, it's really hard to tell what it really is in that sense, because I feel that so the winter survival, it's just not the most simplest trait. It kind of is combined of like being able to survive those low temperature tolerance, being under the ice, being under the water, and also the different, how to say, the variability of high and low temperatures. So yeah, I don't have a good answer for you for that. Yeah.
yeah, I know there is some in barley some very good winter survivorship genetics. I was kind of wondering if you guys were interested in that. Some of that was segregating through the population.
Yeah, that's one of the other projects I'm working on, is just a breeding population to find some more valuable genes with a bigger effects in there. Yes. Yes, Jean-Luc.
You had this slide showing the importance of the time point relative to the heading date.
Yes.
What approach did you use to get those numbers?
So I think, so this is something I just got out of the R package. I can't tell you specifically about the methods, but I think it just uses the magnitude of the coefficient in parametric models. And I think in the random forest model, it uses the number of the node as an average when it's being used in the model through those different cross-validation scenarios. I think this is 100 different iterations I ran with that model to get those values.
Okay, great, thanks.
Yes. Yeah, thank you very much. So the one more question in chat is about if RGB versus multispectral camera were considered for my US data collection. RGB offers much more high resolution, and the color-based vegetation indices are comparable to those developed for satellite imagery, such as NDVI, NDRE. Also, image processing workflows are more straightforward. RGB could be considered, but I just haven't done that because I feel that my multispectral is allowing me to extract the NDVI values faster out of that, more accurately. And the multispectral also allows me to generate the better phenomic relationship matrix, which, in my opinion, is capturing more than just the RGB-based phenomic relationship matrix. Yeah. Thanks.
Hello, everyone. My name is Tim Mulderrig. I'm a second-year student in Mark Sorrells' lab. Happy to be part of the Sorrells lab student seminar extravaganza. I'm excited to tell you guys a little bit more about a project I'm working on, fine mapping a seed-sized QTL on wheat chromosome 2D. This is part of a USDA project that I'm on called the Wheat Coordinated Agriculture Project, or the Wheat CAP, which is a great big collaboration across a lot of wheat breeding programs in the US. So we're interested in wheat because obviously it's a super important crop to a large variety of diets across the world. And thankfully, as we can see, there has been some steady increase in wheat yield in the US over time. And when we consider what grain yield is made of, we're considering it as the interaction of spikes per area on a wheat plant, kernels per spike, and weight per kernel. And that weight per kernel aspect is really what we're interested in. We're thinking about having large seeds with large weight. However, as we know, the world population is really growing. We're going to need lots more food to feed lots more mouths. And unfortunately, across all market classes of wheat, we kind of seen general slight declines in US production over the last 30 years. And so I think it's important for us to consider as we enter into this new wave of increased demand on our food supplies to try to get more genetic information as to what is underlying grain yield, take new approaches, and access some more genetic diversity. So the approach that I am taking towards gaining more genetic information on these yield components is a fine mapping approach. Fine mapping is a process where you have a genomic region where you think there is a quantitative trait locus introducing some variation in a complex trait, and you will determine what are the causal variants within that region that are impacting your trait of interest. And it kind of has a cyclical process, and we're going to go through each of these stages throughout the rest of this presentation. And so my approach to fine mapping has been starting out with utilizing a mapping population. You can see my, oh, you can't see my cursor. Let me get a little laser. utilizing a mapping population and growing out a new generation of that mapping population, hoping to generate crossovers within your region of interest to increase genetic resolution within that region. Then you're going to extract DNA and run a marker panel across that region. And from there, this is kind of like the progeny testing phase, where you are going to grow out different lines for different marker genotypes, phenotype those lines, and do some statistical analysis to try to narrow down your region of interest to get closer to those causal variants. So the mapping population that I'm using is a biparental population generated from the cross of the Opada by synthetic lines. So the synthetic variety is called W7984, and it's the positive allele donor for my population. Synthetic lines I'll talk about a little bit more on the next slide, but they are essentially reconstructed hexaploids created at CIMMYT that were created by doing some genetic work that I'll show, utilizing crossing tetraploid durum wheat to a diverse array of Agelopes tauschii, and that is a wild goat grass species, which is the progenitor for the D subgenome in modern hexaploid bread wheat. And by introgressing this wild D progenitor species, that allows us to access wild germplasm allelic diversity for these yield-related traits. And Opada is another line that's been developed by CIMMYT a few decades ago, and it's a hard red spring wheat. And these parents were chosen because they show variation in kernel morphology, which is our trait of interest, and they have relatively high genetic polymorphism, which is useful for mapping. So thinking a little bit more about these synthetic lines, this is a great figure I stole from a really awesome review paper on CIMMYT synthetics. This right panel shows the evolutionary history of hexaploid bread wheat. We see over the past 0.8 million years, there's been this hexaploid wheat that we grow now has been developed from these ancestral species of the A, B, and D subgenome. And what CIMMYT aimed to do was to reconstruct this hexaploid variety by crossing durum wheat with this really diverse panel of this goat grass species. And then from the crosses, they did embryo rescue and applied a colchicine treatment to double chromosomes to recreate this new primary synthetic hexaploid. They would cross that with a bread wheat variety, and now what's left is a hexaploid line that can be easily crossed with other wheat varieties that has new genetic diversity within the D subgenome. This has been really effective. Over 1,000 synthetic lines have been developed in the last 30 years, and they've been used mainly for the source of wild trait introgression, but they've been also super helpful for the sake of mapping. And they've been using QTL studies to identify traits for all sorts of things: heat stress, stripe rust resistance, all sorts of things. And the specific mapping population that I'm using of the synthetic Opada double haploids and recombinant inbred lines has been a really commonly used mapping population recently, because there's a large amount of available genetic resources, largely created from the WheatCat project. So originally, this QTL was found in a study done by Keith Williams that aimed to map genomic regions across the wheat genome that are impacting all sorts of different kernel morphology traits. And this one on the short arm of chromosome 2D had a pretty high LOD score, and also showed to be stable across different years and growing in the field versus greenhouse, which is why we became super interested in fine mapping this region. And Ellie Toggin, a former student of Mark's, and an undergrad, Elizabeth DeMeyer, worked to narrow down this region to a 13.7 centimorgan range. And they created blood models for this trait and found that it explained 11.4 and 21.7% of variation in 1,000 kernel weight and kernel length. This is still a really large region with over 300 annotated genes across the region. So more fine mapping work is necessary in order to really identify some plausible candidates. So the strategy that we're taking to increase this genetic resolution for purposes of fine mapping was to develop heterogeneous inbred families. So the way that worked is, from the original cross, there were 6 generations of selfing to create recombinant inbred lines. And at that F6 generation, those lines were screened for an SSR marker on chromosome 2D. and lines that were heterozygous and that for that marker were advanced through 3 more generations of marker screens as well, ultimately resulting in F6 derived F10 generation that's heterozygous for a set of flanking CASP markers for that region, which I grew out in the greenhouse last year. And so, yeah, it's pretty intense selection pressure, starting out with over 2,000 real lines. Only 12 of those were heterozygous for our the region we're interested in. But now we've developed it to having 176 HIF families at our current generation. These lines are phenotyped for standard phenotyping methods for 1,000 kernel weight, but the kernel length parameter, we are using a WinSeedle scanner to extract. Basically, you arrange some representative kernels for each line on this image plane, and then you scan the images in the WinSeedle scanner and use image J to extract ellipses values that can tell you things like seed length, width, perimeter, area. And these are the phenotypic values that we're using for our statistical analysis. And the genotyping protocol that I am using is what's called CASP genotyping, or competitive allele specific PCR. This is a PCR fluorescence-based method that each reaction has 2 allele-specific primers that have complementary to specific SNPs at the 3' end, and then a fluorescent tail at the 5' end, and a common reverse primer. And so it allows you to anchor primers to specific subgenomes and polyploid crops, which is super helpful for me, and get good the ability to distinguish between SNP locations really easily. So it's been a great process. Took some trial and error to figure out, but it's been really helpful. And so the general pipeline that I've done to develop new markers is to extract locations of SNPs between my 2 parents of the biparental population across the region using exome capture data that's uploaded on the Tridicy Toolbox database. And from there, I'll gather some flanking sequence from the reference genome and then put those sequences into a software called Polymarker that is used to develop primers for PCR reactions specific for polyploids. And so it utilizes BLAST and a Primer3 software to give me all the necessary primers that I'll need for my CASP reaction. I'll order those primers and validate them to make sure they're properly segregating on the parent DNA. All sorts of issues can come into play when you're working with polyploids of non-specific binding or whatever. And so this has taken some trial and error, but we've been able to develop a pretty dense marker panel to use. Some of the early results that I'm getting for this genotyping, this figure in the top left is showing, first I did a marker screen of all the F6 derived F10s progeny for 2 flanking markers for the sake of wanting to grow out different lines of different genotypes in the field for phenotype analysis this summer. And so you can see most of the lines are going to be homozygous across the whole region or heterozygous across the whole region. We're more interested in these lines at the bottom that show evidence of a crossover, because that can give us genetic resolution to make some statistical inference. And so I'm going to grow out about 600 lines that have shown evidence of a crossover with some parent checks in head rows this upcoming field season. The goal is eventually to have genotype all these lines for a more dense marker panel. Here, I've started this process genotyping 4 markers across the region, and that will give us obviously even more genetic resolution for our analysis down line. And this is an example of what an output from running a plate of CASP would look like. You can see that the different lines cluster in different regions if they are homozygous for one of the alleles, homozygous for the other allele, or heterozygous. And I've been doing some manual adjustment for my calls as well. We're also utilizing RNA-seq to identify some plausible candidates within the region. From last field season, I grew out some head rows of the 2 parents and a few of my HIF lines and gathered heads across different time points during grain fill. And we're going to extract RNA from these heads and run some RNA-seq, hoping to get some genes that are expressed specifically in the grain, which we would think would be plausible candidates. Also genes that are differentially expressed between our 2 parents, and see if there's any genes with interesting expression profiles across grain fill that could be plausible for a kernel length trait. And ultimately, our goal is to do some candidate gene verification and positional-based cloning. once we have some good candidate genes for them, this region will compare it to annotated genes that are for the reference genome. There's been papers citing all sorts of pathways relevant for seed size, protein degradation, cell division and expansion, starch and protein, and with content within the grain. So those will be things I'll be on the lookout for as well. And then I'll ultimately probably use a VIGS approach for candidate gene validation before doing cloning. Wanted to acknowledge people who have helped me along the way, all my advisor, Mark Sorrells, and all the people from the Sorrells lab, past and present. Special shout out goes to Ellie Toggin, who started this project and has been a really great mentor and resource for me as I've navigated fine mapping. Also, thanks to my committee, the members of the JS group, and to my funders. Happy to take any questions as well. Ed.
Yeah, I was kind of wondering on the positional cloning. It looked like you were looking at a large number of HIP families rather than focusing in on just one or two and then going massive on that. What was the strategy on that, going with large numbers of HIP families rather than just focusing in on the one?
Yeah, the question was for eventual positional-based cloning. It seems this year I'd focus on a lot of HIP families instead of just a few. Why was that choice made? When I grabbed that previous generation, I wasn't really considering it super closely, and I'm planning for the next round to focus on just a few families to increase that resolution, because it'll probably take at least another round of this cycle to get to that point.
Can you see the difference on a single plant?
I can. I think I showed a slide earlier that had a picture of the two parents. I don't know if I pointed it out. There are some thrushability issues with synthetic lines, so sometimes it can be a little complex. So I want to make sure I'm perfecting my seed cleaning process before doing the image analysis. But sometimes you definitely can see distinct differences between the kernel shapes between the two. Junk.
I mean, not to fight with Ed, but he sort of said, why not focus on few or maybe just a single family? But what would I mean, I can see an advantage of focusing on several families, which is you might be able to find some interaction between a particular allele and the genetic background, right? You only have one background, which you would not have in one family. You wouldn't find that. Now, looking for that has trade-offs, I suppose. But anyway, say more about this process.
Jean-Luc's asking kind of if there is value in screening a lot of HIP families instead of just a few. And I think it's a good point, something I want to consider more, especially, yeah, is there information that can be gained from running like a background marker say on these to see if there is some other interactions that I'm not considering? I think, yeah, there's definitely more to be explored there to make sure that the next cycle I do is as effective as possible. Yeah.
You're planting RNA-seq. What tissue are you getting?
The question is, what tissue am I using for RNA-seq? I collected heads, and I'm going to use grain tissue from the seeds that grew within the middle of each head. Yeah, so RNA-seq can be hard for starchy tissues, but hopefully using like I found some references for a good protocol that can account for that.
What part of the grain specifically are you aiming for, the endosperm, or how is the tissue specific in the grain?
Ask them one more time, sorry.
How is the tissue specific inside the grain? Are you aiming to extract the endosperm, or?
The question is, how specific within the grain am I aiming to look? Am I just looking at the endosperm? I think I'm just going to look at the entirety of the grain. Yeah, obviously mostly endosperm, but break down the whole thing and extract from the whole part. Well, thanks for listening.
All right, so only 10 more minutes of grains and then we'll let you guys go. All right, so I'm Miranda, and today I'm going to be talking to you guys about exploring the landscape of organic small grains cultivation in New York. So definitely a change up from the last couple of presentations. So as I'm sure most of you are aware of, there's been a huge trend in the last several years of consumer and market interest around locally and organically grown specialty crops. While this trend has definitely encompassed more fruit and vegetable crops, over the last several years, it's also grown to include small grains. So one class of small grains that is getting more interest and more traction in markets is something called ancient grains. So for those who aren't aware, ancient grains are not something like quinoa. We definitely define it a little more specifically in our lab as ancestors of modern wheat varieties. So the 3 species that we work with are emmer, einkorn, and spelt. So why are consumers getting more interested in these grains? There's a couple main reasons. First and foremost, they're interesting, right? So they have a distinctive and unique flavor when compared to modern wheat varieties. So people like artisan bakers and chefs are really interested in including these grains in their businesses just because they're different. Also, emmer and einkorn have been found to have a lower gluten content compared to modern wheat varieties. So this means that people who have sensitivities to gluten may be able to digest these grains more easily. So that's another reason for their more recent popularity. So as you can imagine, since consumers are looking for grains, specialty crops like this that are grown locally and possibly under organic management, we're also seeing a trend of growers who are more interested in adopting these varieties into their programs. Unfortunately, with these grains specifically, there are some challenges to growing them here in New York. The 2 main challenges that I'm going to be talking a little bit about today, first and foremost, these grains are not well suited to production by small farms. And the main reason for this is that most varieties of these grains are covered varieties. So that means that the hull does not separate easily from the kernel, and it needs to undergo a process called threshing, which is really labor intensive, time consuming, and really difficult for small farmers to be able to do if they don't have the resources in order to commit to that. So as you can imagine, this is a really big barrier for adoption of these grains for small farms. Additionally, another big barrier for these grains is that they just don't really grow well in the New York environment. They aren't particularly compatible to our region. So given these problems, our 2 big goals for breeding ancient grains are to develop varieties that are free threshing, as well as varieties that are well adapted to our region. So to start with the free threshing trait, the development of these varieties was initiated several years ago by a previous technician in our lab, James Tanaka. He began by making crosses between covered and free threshing parents, or in the case of einkorn, partially free threshing parents. As it stands, we have 3 populations that James began of spring, emmer, and einkorn, and a winter type spelt. So the most advanced populations that we've been working with are the spring, emmer, and einkorn. As the einkorn stands, we are still in the process of increasing the number of seed we have so that we can evaluate it and make good selections. The spring emmer is our most advanced line, and we've had a lot of success with it. We've had a couple of years of preliminary yield trials, and we're really close to a varietal release. Most of the emmers thresh out fully, which is really exciting. And I have a nice example here to the right, where I'm comparing at the top a covered parent of emmer, and the bottom is one of our free threshing varieties. And I just took this picture a few weeks ago from our last year's trial. And also exciting that most of these lines that are our best performers are competitive in terms of yield with their parents as well. So the second problem that we're addressing for regional adaptation is a problem that I've helped initiate the solution to. Last year, I began to do crosses with emmer and einkorn to develop winter-tolerant populations. So we began by making selections of some free threshing springs of each of these grains from our developed populations that I just described, and I crossed these with some covered winter parents. So the big goal here is to be able to transfer the free threshing trait into the winter background. Of course, I just made these crosses last year, so we're still in the early stages of advancing these generations, and we're really looking forward to getting them out into the field to see how they do in our climate and how that threshability trait has held up through the crosses. And I have on the left there a picture of one of the emmer heads that I had emasculated, so really fun to work with, super tiny, but it was a good experience for sure. So as you probably guessed, some of the big picture goals that we're trying to reach here with this breeding program is really to support farmers by increasing accessibility and profitability of growing these grains in New York so that they can have access to this market. One of the questions that I've had since I started the program is, we know that these 2 traits that we're working on are super important to be able to grow them here and grow them successfully. But is there anything else that these growers will really care about before they decide to actually adopt these grains into their operations? Some previous work towards this has been done. A previous graduate student in our lab performed a survey where she was asking farmers in New York who grew wheat organically what their trait preferences were, and she found some really great results. I really want to know if these preferences hold true for ancient grains as well, since these are 2 different styles of growing and management. So this leads me to one of my research projects, which is a survey of organic small grains growers here in New York State. Some of the main research goals for this project are to assess interest and current involvement in growing ancient grains from growers specifically in New York. We want to understand what kind of choices that they're making around either adopting or choosing to not adopt these types of grains. And of course, we want to understand what type of trait priorities that are specific to ancient grains do these farmers really care about. And then finally, we also want to take this opportunity while we're surveying growers to explore the demographics of organic small grains growers in the state, since there isn't a lot of information out there about that at this time. So the sampling approach is fairly straightforward. We have a few basic selection criteria for our grower participants. Of course, we want them to be based in New York State, and we want them to be actively growing small grains on their New York farms, specifically for seed. And then we either want them to be certified organic or follow organic management practices. So snowball sampling is the approach that we chose in order to try to reach as many growers as we possibly can. We're beginning with just a few initial contacts, such as Cornell Cooperative Extension, grower groups that are involved in small grains production, as well as self-identifying organizations. That way we can reach growers from underrepresented groups as best as we possibly can. And finally, after our first wave of contacting growers, we'll be asking respondents to help identify other potential participants within their networks that may be eligible to be included in our survey as well. And I just wanted to share with you a tentative survey design to kind of show how it is that we'll be approaching and asking these farmers our questions. So this will be a web-based branching style survey with mostly multiple choice questions. One example of a question that we'd like to ask is, in the past year, including the current season, have you grown or considered growing any ancient grains? And depending on what their answers are to one question, they'll be given a different set of questions to therefore answer that is relevant to their previous response. So in this example, we would be curious to understand why growers are interested in growing it, whether they have grown small grains, and if they haven't, what concerns or barriers do they perceive around growing these grains? At the end of the survey, of course, we'd like to look at trait preferences. So we're interested in giving a few traits for growers to rank by their priority. And then finally, demographic information at the very end. So some main anticipated outcomes for this research. At the very start, we'll be curating a list of growers in New York State. And this is really valuable because this could be a really good potential resource for future surveys that go more in depth, as well as for participatory work in the future. Of course, we'll be walking away from the survey, hopefully with a strong assessment of grower interest, motivation, and current involvement with ancient grains in New York State, as well as their trait priorities. And hopefully, if people are comfortable answering demographic questions, we'll have a better understanding of diversity and social identities that help make up the small grain grower population in the state. And then, of course, overall, one of our main goals from the survey is that we'll come away from it with really good information about these grower preferences that can help inform some of the decisions that we make in our breeding program so that we can best support the farmers that we want to. So I'd like to end with thanking, of course, my committee members, first and foremost, Mark Sorrells, Holly Tufan, and Vipen Kumar for all of their support in developing this research. And of course, a big thank you to everyone in the Sorrells lab, past and present, for their support and help. And I'd like to give a special thanks to everyone in the lab of John Luke, who also gave me feedback on this work recently. And then, of course, thanks to my sources of funding through USDA and NEPA. And thank you all for listening to my talk today, and I'm happy to take questions. Thanks.
Do you see working with this panel of growers mostly to set the priorities, or will you also just working with them farther along the process in certain people?
Right. So the question was, do we see working with this panel of growers in order to initially set the priorities for the breeding program or to continue working with them in the future? I think the answer is kind of both. So we already have our main priorities with this breeding program being the threshability trait and transferring to a winter background. So we want it to be able to be grown here well. But it would be great if for some other traits, something like lodging, disease resistance, flowering time, if we could know that some growers really prefer that, those that are likely to pick up these traits, then now is the really great time, since we're just beginning some of these populations, to be able to select for those early on. But additionally, if in the future we have the opportunity to include these growers in participatory trials, that would also be excellent, of course. Thank you.
So you mentioned there was maybe a survey for wheat. I was wondering if the traits that you present for them to be able to choose from, if they're all the exact same traits that were listed on the previous survey, or if there are new ones that are specific to ancient grains.
Right. Great question. So the question was if the traits that we'll be including in our new survey will be derived from the previous work that I had mentioned, or if they're more specific to ancient grains. As it stands, we're still finalizing what those traits will be. Currently, I'm leaning towards including the ones that were prioritized in that previous study that were for wheat. There's a lot of overlap. So most likely we'll be going with maybe the top 5 or 6 traits that were prioritized by the wheat growers.
We were talking about regional adaptation. I heard you talk about winter survival as a priority. Are there other characteristics that you think would influence regional adaptation? They're going to be influencing others.
So the question was, do we think there are other traits or aspects that can help influence regional adaptation? Absolutely there are. And I can give you one example. We had some grower feedback at a conference last year that they have some issue growing spelt in New York where they'll have some early freezes that'll lessen their yield because it can kill off the flower. So something like that can help inform if we want to select for a later heading date. So absolutely, there are a lot of other aspects that will influence whether or not these grains will grow here. It's really complex for sure. And winter survival is just one aspect of it. Thank you. Okay, thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.